made up from phospholipids, a macromolecule which is built out of a glycerol head and two fatty acid tails. This construction makes the phospholipid amphipathic, making it have a hydrophilic head and two hydrophobic tails. These crucial components come together to form what we know as the phospholipid bilayer. Embedded within this phospholipid bilayer are proteins which have various functions, some which will be discussed later. Diffusion is one of the basic ways that substances may get into a cell. Diffusion is the movement of molecules of any substance from a higher concentration to a lower one. This occurs naturally as molecules diffuse down their concentration gradient till an equilibrium is formed. Nonpolar molecules such as hydrocarbons, carbon dioxides, and oxygen can easily pass through the membrane. But the hydrophobic interior of the lipid bilayer hinders the transit of polar molecules, which are hydrophilic. Osmosis is practically the same process, but it has to do exclusively with water. Due to its chemical buildup, water is polar and thus cannot diffuse through the bilayer. This is where proteins start to play an important role. A special protein called an aquaporine creates a tunnel which is hydrophilic inside, allowing the water to easily pass in and out of the cell, moving down its concentration gradient. However, the strive for equilibrium in some cases can destroy the cell. A hypotonic environment hosts a solution that is less concentrated on the outside of the cell than on the inside. Due to water trying to establish an equilibrium and moving down its concentration gradient, water will flood the cell and the cell will lyse. On the other end of the spectrum is a hypertonic environment, one which has a higher concentration on the outside of the cell than the inside, forcing water to attempt to dilute the outside concentration, thus draining the cell of its water. In this example, the cell is shown. Within these environments, cells cannot function properly, but in an isotonic solution, which has the same concentration as the cell, thus water flows in and out at the same rate, keeping the cell in a healthy shape. Diffusion and osmosis happen naturally, thus there is no need for energy to be used up. Facilitated diffusion, on the other hand, is the use of proteins, which are embedded in the phospholipid bilayer to help polar substances cross into the cell. There are three types of these proteins: ion channels, gated ion channels, and carrier proteins. Ion channels provide a hydrophilic tunnel for substances which are polar. In fact, we have already looked at one of these ion channels earlier. An aquaporine is an example of an ion channel. Then a variation of the first comes the gated ion channel. These work just like the ion channels, except they have lids and require a certain substance to bond for the gate to open. Third, we come to the carrier proteins. These are more complex than the others due to the fact they change shape to allow passage for substances. These proteins also require energy in the form of ATP. Why? Because these proteins can pump against the concentration gradient, taking in more of one substance than what would naturally diffuse into the cell. One example of a carrier protein is the sodium-potassium pump. This specialized protein takes in potassium while pumping out sodium. In its original shape, it has a higher affinity for sodium. Once full, a phosphate group activates the protein, which changes shape and releases the sodium side. While in this state, the protein has a high affinity for potassium, thus attracting it. Once full of potassium, the phosphate group is released and the protein changes to its original shape, thus releasing the potassium inside of the cell. Endocytosis is the process of a cell taking in molecules and other biological matter by forming vacuoles around the targeted substances. Phagocytosis is the process where a cell will engulf a food particle by extending the cell membrane around the substance. Penocytosis is when a cell forms a vesicle by sinking part of the cellular membrane in, allowing fluid to flow in. The fluid is not what the cell is looking for. The cell is actually gathering the molecules that are dissolved in the fluid for later use. Receptor-mediated endocytosis is the bulk gathering for certain substances. Proteins which are embedded in the cellular membrane may contain specific receptor sites to which only certain substances will bond. Receptor proteins cluster together in a region called coated pits. Coated pits get their name from the coproteins which line the outside of the vesicle. Finally, we come to the process called exocytosis. This is where a vacuole containing waste will fuse to the cellular membrane and expel the waste into the extracellular fluid.